In this module, we will discuss dimension reduction. As a reminder, we have already discussed the preprocessing methods including image analysis for array-based data and alignment for RNA-seq-based data, normalization, and expression summaries. Once we have expression summaries, we then perform our planned statistical analysis. We will now focus on our last topic, dimension reduction. Rudimentary exploratory procedures are quite helpful in understanding the complex nature of multivariate relationships. Graphical procedures for depicting high-dimensional observations in a few, such as 2 or possibly 3, dimensions is one exploratory procedure. In order to display high-throughput genomic data, which includes thousands of features, the dimension of the data must first be reduced. So one clear benefit of dimension reduction is better visualization. That is, if we reduce our gene expression dataset from thousands of features to 2 features, we can produce a scatter plot of those 2 features to visualize the relationship between observations. If we reduce the thousands of genes to 3 features, we can produce a 3-dimensional scatter plot using specialized graphical routines and rotate the plot to observe the relationships among 3 variables. We can use a different plotting symbol to indicate a nominal class variable so we can visualize the relationships among up to 4 variables. However, beyond 3 or 4 variables, we lose the ability to graphically depict the data. Nevertheless, when you have a high-dimensional feature space, such as with high-throughput genomic data, which includes thousands of features, the dimension of the data must first be reduced if you want to visually examine the relationship between observations. Dimension reduction methods are quite frequently used in the analysis of single-cell sequencing data because often researchers are interested in identifying genes uniquely expressed in different cell types. In this module, we will cover some commonly used dimension reduction methods to reduce thousands of features to only a few features, say 2 or 3. Thereafter, we can construct graphs that allow us to visualize the data. Another added benefit of dimension reduction is that we can use the reduced features in a statistical analysis, such as predictive modeling, which, because there are now only a few features, the analysis will be faster. Also, the reduced features are combinations of the original variables, and little to no weight is placed on noisy features. Therefore, dimension reduction methods usually remove noisy or extraneous features from the dataset. A common use of dimension reduction methods is to examine a 2-dimensional scatter plot using a different plotting symbol to indicate batch, in which the samples were run. This allows the investigators to determine if there are unmeasured batch effects. In the end, the goal of dimension reduction is to reduce the number of variables of a dataset while preserving as much information as possible. As discussed, some benefits of reducing the dimension of a large dataset include that smaller datasets lead to better visualization, faster downstream analysis, can remove noisy or extraneous features, and can uncover unmeasured batch effects. In summary, the overall goal of dimension reduction is to reduce the number of variables of a dataset while preserving as much information as possible. Dimension reduction methods can generally be separated into 2 categories, global and local methods. Global methods include principal component analysis and multidimensional scaling, and aim to preserve relative distances or rank information between points, resulting in a stronger focus on the preservation of distances between points that are farther away from each other. That is, global structure preservation means that the relative positions between clusters are preserved, such that because global structure focuses on relationships between clusters, a dimension reduction with a high-quality global structure preservation could help us understand, for instance, the degree of similarity between different clusters. We will also include Fisher's discriminants as a method falling into the global dimension reduction method framework. Local methods are more commonly used for direct visualization of pre-processed biological data, and more recently developed local methods that are now commonly used include T-distributed stochastic neighborhood embedding, or TISNE, and uniform manifold approximation and projection, or UMAP. Local structure methods aim mainly to preserve the set of high-dimensional neighbors for each point when reducing to lower dimensions, as well as the relative distances or rank information among neighbors. Local preservation means that neighbors in high-dimensional space should still be neighbors in low-dimensional space. These local methods aim to preserve cluster structure, though they may fail to preserve the overall layout of clusters in space. First, we will discuss global methods, starting with principal component analysis. Principal components are new variables that are constructed as linear combinations of the initial variables. These combinations are constructed such that the new variables, that is, the principal components, are uncorrelated, and most of the information within the original variables is squeezed or compressed into the first components. So, although with p-dimensional data you will have p principal components, principal component analysis tries to put the maximum possible information into the first component, and then the maximum remaining information into the second component, and so on, as shown in this plot. Therefore, often the components with low information are discarded, and the remaining components are considered to be your new variables. Organizing the original data into principal components will allow you to reduce the dimensionality without losing much information. As mentioned, principal component analysis, or PCA, projects high-dimensional data to a lower dimensional space, which maximizes the variance of the projected data. First, the variables are standardized so that each variable contributes equally to the analysis. This is because if there are large differences between the ranges of variables, those variables with larger ranges will dominate over those with smaller ranges. For example, a variable like age may range from 0 to 100, and it will dominate over a variable like hemoglobin that ranges between 12 and 18 in normal healthy subjects, which may lead to biased results. Next, the covariance matrix is calculated. This is calculated because the aim is to understand how the variables vary from the mean with respect to each other, or in other words, to see if there is any relationship between them. Sometimes variables are highly correlated such that they contain redundant information. Highly correlated variables can be identified from the covariance matrix. The sign of the entries in the covariance matrix matter. If positive, then the two variables increase or decrease together. If negative, then while one variable increases, the other variable decreases, so they are inversely correlated. Next, compute the eigenvectors and eigenvalues of the covariance matrix. These are linear algebra concepts that we compute from the covariance matrix in order to determine the principal components of the data. Eigenvectors of the covariance matrix are actually the directions of the axes where there is the most variance or most information, and we call these principal components. Their corresponding eigenvalues are simply the coefficients attached to the eigenvectors, which give the amount of variance carried in each principal component. By ranking your eigenvectors in order by their eigenvalues, highest to lowest, you get the principal components in order of importance. For those interested, the first principal component is the line that goes through the origin in which the projection of the points is the most spread out. Or mathematically speaking, it is the line that maximizes the variance, the average of the squared distances from the projected points to the origin. The second principal component is calculated in the same way with the condition that it is uncorrelated, that is, it is perpendicular to the first principal component, and that it accounts for the next highest variance. This continues until a total of p principal components have been calculated, equal to the original number of variables. An important thing to realize here is that the principal components are less interpretable and don't have any real meaning since they are constructed as linear combinations of the initial variables. Dimension reduction is then performed by selecting a subset smaller than p among the p principal components. This is usually accomplished by either calculating the percentage of variance explained by each principal component and selecting some k that is less than p at which an elbow occurs, or retaining the first k principal components that explain a predetermined percentage of variance explained in the data, such as 80%. Note that when applying dimension reduction methods such as PCA to high-throughput gene expression data, we can either treat the subjects as data and the genes as features, or treat the genes as data and the subjects as features. The choice of whether you reduce the dimensionality with respect to genes or subjects is determined by whether you want to understand the relationship among either genes or among subjects. Let's look at an example. First, download the Cancer Genome Atlas breast cancer dataset using the TCGA BioLinks bioconductor package using this syntax. Because this is a large dataset and takes a while to download, this last call saves the workspace such that if you close R, you can simply load this workspace rather than re-executing these lines to download these data. Suppose we are interested in analyzing the samples from subjects whose sample was from either a primary tumor or solid normal tissue. We can extract that subset of subjects using this function. Doing so, we see that there are 1,111 primary tumors and 113 solid normal tissue samples. Now we transform the count data and then filter the gene expression data matrix, so there are now 19,972 genes. Prior to performing principal component analysis, we standardize the data matrix. The PRComp function performs principal component analysis. We can apply the PRComp function to treat the genes as data and the subjects as features, or we can apply the PRComp function to treat the subjects as data and the genes as features. The scree plot is a plot of the percentage of variance explained by each principal component, sorted in order from largest to smallest. You can see that most of the variance when treating the genes as data and the subjects as features can be explained by only one principal component, while the first four principal components treating subjects as data and the genes as features only explains around 30% of the variance. Typically, we use this information to identify how many low-dimensional features to use. Ideally, we keep the number of low-dimensional features, which are linear combinations for PCA, that explain 80% of the variability. While the scree plot on the previous slide shows that we can effectively reduce the number of genes to one linear combination to preserve over 80% of the variability, here we are examining a scatter plot of the first and second principal components, simply because two-dimensional plots are more interesting than box plots. And this scatter plot reveals that the classes primary tumor and solid normal tissue generally have different expression patterns. Therefore, we can reduce the gene expression features to one or at most two principal components and retain most of the information that is useful for identifying these two classes of patients. Multidimensional scaling is another dimension reduction technique. For n data points, we calculate a distance matrix D, which stores the distance between every pair of n items. The goal is then to find a representation of the items in few dimensions such that the inter-item distances nearly match the original distances. It may not be possible to match exactly the ordering of the original distances. Consequently, scaling techniques attempt to find configurations in k less than n-1 dimensions such that the match is as close as possible. In classical multidimensional scaling, Euclidean distance is used. The solution is not unique because the Euclidean norm is invariant to shifting and scaling. Therefore, Procrustes analysis, which rotates and stretches or shrinks, can be applied. Classical multidimensional scaling proceeds by setting up the squared distance matrix. Then apply double centering using this matrix calculation, where C is the centering matrix, where n is the number of data points, I is the n by n identity matrix, and J sub n is the n by n matrix of all ones. Then the k largest eigenvalues and corresponding eigenvectors of B are determined, where k is the number of dimensions desired for the output. Then the new X matrix is obtained using this matrix calculation, where U sub k is the matrix of k eigenvectors and lambda sub k is the diagonal matrix of the k eigenvalues of B. Suppose for our breast cancer data set, we want to perform multidimensional scaling using the samples as data and genes as variables. The CMD scale function performs classical multidimensional scaling of a data matrix. Applying the CMD scale function to our data, where we calculated Euclidean distance as the distance between the samples and seek to represent our data in 2 dimensions, we see the resulting scatter plot. As mentioned, multidimensional scaling is equivalent to PCA if Euclidean distance is used. Also, as mentioned, the solution is not unique because the Euclidean norm is invariant to shifting and scaling. Therefore, Procrustes analysis, which rotates and stretches or shrinks, can be applied. If you look back at the principal component scatter plot and compare this scatter plot for multidimensional scaling, you should notice that they are mirror images of one another. Here we perform multidimensional scaling using the samples as data and genes as variables, but this time we calculated Manhattan distance as the distance between the samples. The resulting scatter plot of our lower dimensional data set differs using Manhattan distance, but we still see separation between our 2 classes. Principal component analysis and multidimensional scaling are unsupervised. That is, no phenotypic information or class labels were used to reduce the dimension of the data. Instead, the reduced number of dimensions k are selected to maximize the variance explained by the variables. Alternatively, Fisher's discriminant analysis is a supervised dimension reduction method that seeks to maximize the difference between the observed groups. Suppose that there are j groups in the data set. That is, there is a nominal phenotypic variable that takes on j levels. The mean vector of a specific group is denoted by mu sub j. Averaging these j mean vectors yields the mean vector of the combined groups. The between group variance covariance matrix can then be estimated using this equation, as well as the within groups variance covariance matrix can be estimated using this equation. Fisher discriminants are then the eigenvectors and eigenvalues from the eigendecomposition of the between to within variance covariance. Notice that for j groups, there are at most j-1 eigenvectors. If j is large, a smaller number could be selected for reducing the dimensionality. The resulting eigenvectors are scaled. The linear combination associated with the first eigenvector is the sample first discriminant. If there are more than 2 groups, such that j is greater than 2, the second linear combination associated with the second eigenvector is the sample second discriminant. Because the variance covariance matrices must be a full rank, we require that the number of samples exceed the number of variables. Therefore, we more aggressively filtered the breast cancer data set so that now there are 124 variables and 1,224 subjects. Previously, we used the LDA function in the MassR package for fitting a linear discriminant analysis. In that module, I did not describe all of the components of the output object as we focused on prediction in that module. Here you can see the various objects stored in the LDA fitted object. The scaling object returns the Fisher discriminants. Because there are 2 classes, that is, j equals 2, we only have 1 Fisher discriminant. You can see the scaling of the individual genes. The box plot demonstrates that gene expression data can be condensed into one variable that is a linear combination of the 124 genes that separates the 2 groups well. So Fisher's discriminant analysis is a dimension reduction method, but it differs from others in that first, Fisher's discriminant analysis is a supervised dimension reduction method that seeks to maximize the difference between the observed groups. Second, when there are j groups, there are at most j-1 eigenvectors. So in our example, there were only 2 groups, so only 1 Fisher discriminant is available. Because you cannot produce a scatter plot with 1 vector, we looked at box plots instead. And third, Fisher's discriminant analysis requires that the number of samples exceeds the number of variables because the inverse of the variance-covariance matrix is part of the calculation. Stochastic neighborhood embedding was a method developed in 2003 which converts Euclidean distances between samples into conditional probabilities that represent similarities. It creates a low-dimensional embedding by enforcing the conditional probabilities in lower dimension to be similar to the conditional probabilities in the higher dimension. There were different improvements to the SNE method, including T-distributed stochastic neighborhood embedding, or TSNE, which uses the long-tail T-distribution rather than a Gaussian distribution. Therefore, TSNE is a nonlinear dimension reduction technique for reducing high-dimensional data to 2 or 3-dimensional points such that there is a high probability that similar objects will have point near one another and dissimilar objects will have distant points. Suppose we have n high-dimensional observations. For example, n subjects each having thousands of gene expression measurements. Consider two subjects I and j. We define the conditional probability of j given I. That is, for any two objects I not equal to j, the similarity of object x sub j to x sub I is the conditional probability p of j given I. That x sub I would pick x sub j as its neighbor if neighbors were picked in proportion to their probability density under a Gaussian kernel centered at x sub I. We then define the similarities using this equation, which is motivated because p sub I and p sub j from the n samples are estimated as 1 over n. So the conditional probability can be written like this. The bandwidth of the Gaussian kernel is sigma sub I, which is adapted to the density of the data. TSNE aims to learn a low-dimensional map in lower dimensional space that reflects the probabilities p sub ij as well as possible, where the number of dimensions is typically 2 or 3. Specifically, we define the lower dimensional probability q sub ij using this equation and set q sub ii equal to 0. A heavy-tailed student t distribution with 1 degree of freedom, which is the same as the Cauchy distribution, is used to measure similarities between low-dimensional points in order to allow dissimilar objects to be modeled far apart in the map. The locations of the lower dimensional points y are initialized using a multivariate normal distribution, then updated by optimizing this equation, such that the locations of the points in the lower dimensional space are determined by minimizing the Kahl-Black-Leibler divergence of the distribution P from the distribution Q. This minimization of the Kahl-Black-Leibler divergence with respect to y is performed using gradient descent. The result of this optimization is a lower dimensional set of features that reflects the similarities between the high dimensional inputs. The R-TSNE R package includes the R-TSNE function, which can be applied to our high-dimensional gene expression data set. On the left is the scatter plot of the TSNE result. Comparing to the principal component plot on the right, it appears that TSNE works better because there is more separation between our two groups, primary tumor and solid normal tissue. TSNE is considered as a nonlinear machine learning algorithm and performs better than traditional linear dimension reduction algorithms, such as PCA and MDS in many applications, including genomic studies, especially when the goal is to find local structure. For that reason, TSNE is one of the standard dimension reduction methods for single-cell RNA-seq data. TSNE is not only a visualization tool, but the resulting lower dimensional features may be used in downstream statistical analysis, including clustering and classification. UMAP was developed to overcome some perceived shortcomings of TSNE. The UMAP algorithm consists of two primary phases: graph construction for the high-dimensional space and optimization of the low-dimensional graph layout. The graph construction phase is as follows. For each observation, find the k nearest neighbors using a given distance metric. Usually Euclidean distance is used. Then, for each observation, compute rho sub I, which is the smallest distance from observation I to a neighbor. Also compute a scaling factor sigma I by solving this equation, where again k is the number of nearest neighbors. Then define the weight function using this equation. Now you can construct a weighted graph using the weights as defined in this equation, where for each edge, it holds that x sub I is the nearest neighbor of x sub j. Then to optimize the low-dimensional graph layout, we first initialize the low-dimensional object y using spectral embedding, then iterate over the graph edges in G, applying gradient descent steps on observation I on every edge, which applies an attractive force in the low-dimensional space on observation I towards observation J. Recall, J is I's nearest neighbor, so we want to move points in low-dimensional space closer when they are close to each other in high-dimensional space. Then apply a repulsive force to observation I, pushing it away from observation L, where L is chosen by randomly sampling non-neighboring vertices. This repulsive force pushes apart points in low-dimensional space that are further away from each other in the high-dimensional space. The equations for the gradient approximations used in the optimizations are omitted but can be found in this reference. The official implementation of UMAP is available through a Python package. Here, the UMAP-R package is described. First, load the UMAP package. Then apply the UMAP transformation. The output is an object, and we can get a summary of its contents by printing it. The main component of the object is layout, which holds a matrix with the low-dimensional coordinates. These coordinates can be used to visualize the dataset. We applied UMAP to the TCGA dataset using the default settings, which are listed here. For a description of each field, see the documentation in help umap.defaults or the original publication. To create a custom configuration, we can make a copy of the default object and then update its fields. To perform the UMAP projection with these settings, we can run the projection again and provide the configuration output as a second argument. Another way to customize the algorithm is to specify the parameter values one by one as arguments to the function call. This command achieves equivalent results. On the left is the UMAP using their default parameters. On the right is the UMAP using the custom parameters. To summarize, the dimension reduction methods covered in this module were principal component analysis, multidimensional scaling, Fisher's discriminants, t-SNE, and UMAP. PCA and MDS are both based on eigenvectors and eigenvalues of the covariance matrix and are equivalent when Euclidean distance is used as the metric distance and result in linear combinations of the original variables. On the other hand, t-SNE is a more recently developed dimension reduction method and results in a nonlinear combination of the original variables. Because it is nonlinear, it usually performs better than PCA and MDS. These first three methods are unsupervised. That is, no phenotypic information or class labels were used to reduce the dimensionality of the data. The same holds for UMAP. Alternatively, Fisher discriminant analysis is a supervised dimension reduction method that seeks to maximize the difference between observed groups. Unlike PCA, MDS, t-SNE, and UMAP, where we can specify how many features we want to keep, here are some references.